The human body consists of trillions of specialized cells that control cell division and growth. If there were to be a genetic change in any of these cells, there would be uncontrollable growth and division. For there to be this, there would need to be genetic change in 3 types of genes. These genes being proto-oncogenes, which are responsible for cell division and growth, tumor suppressor genes, which control cell division by halting it, and DNA repair genes, which would preserve genetic codes. In attempts to treat this disease, scientists grow human cells in a lab to study how they function and test new treatments as well. For this to be possible, however, there would need to be huge populations of identical cells that would survive for years. But cell lines would eventually die, in days. Fortunately, a specific biologist named George Guy was given an abnormal cell that was purple, shiny, and jelly-like. In this cell, cells in it would divide chronically. Each time a cell would die or undergo apoptosis, a new cell would take its place and thrive. The first HeLa cell was discovered, named HeLa after the patient with the immortal tumor Henrietta Lacks. This cell was extracted a few months after Henrietta Lacks' death due to cervical cancer. Dr. George Guy discovered that these cells would double every 24 hours in size. After the studies and tests, these cells were distributed across the world to anyone desiring to do tests on them for free. However, Henrietta's family had not been informed of their distribution and their extraction. How these cells were immortal, however, had not remained clear. The performance can be explained by the expression of an overactive telomerase, an enzyme that rebuilds telomeres after each division, preventing cellular aging, cellular senescence, and allowing perpetual divisions of the cells, thus preventing apoptosis or cell death. HeLa cells also tend to increase in cell count the more time they have. HeLa cells also provide the most scientific breakthroughs, including treating diseases. The HeLa cell line gave scientists plenty of time and possibility to conduct repeatable experiments on human cells, without testing directly on humans as well, and to this day, HeLa cells have also saved countless of lives and many scientific landmarks such as cloning, gene mapping, and breeding fertilization, and the polio vaccine. Have used all HeLa cells and owe everything to the life and death of Henrietta Lacks. In the early 50s, when the polio disease was at its peak, HeLa cells was actually essential for the vaccine, since it could be infected and replicate the virus, thus giving scientists what they needed for vaccine testing and studies. They've also been known for various diseases including mumps, measles, HIV, and Ebola, which have been studied by the HeLa cells. HeLa cells, like many tumors, have error-filled genomes. With 1 or more copies of many chromosomes, a normal cell has 46, whereas the HeLa cell has 76 to 80, 25 of which have been heavily mutated. HeLa cells were also the first to be cloned. They've been taken to outer space, and the enzyme telomerase is an enzyme that prevents cell death via division, was also found in HeLa cells, as well as the discovery of the main cervicular cancer, which was the reason of Henrietta's death. It was because of HPV, and because of this discovery, there's also been a vaccine for cervicular cancer. The amount of scientific breakthroughs caused by HeLa is truly remarkable and uncountable. They're capable of traveling on the surface, including human skin and lint, and capable of invading cultures of other cells, including rapid division. All this came from Henrietta's Lacks, the woman of immortality.